Dear learners, in this video we are going to study chapter 3 of class 11th biology NCERT, that is plant kingdom. Okay, so here we start. In the previous chapter we looked at the broad classification of living organisms under the system proposed by Whittaker in 1969, wherein he suggested the 5 kingdom classification with Monera, Protista, Fungi, Animalia and Plantae. In this chapter we will deal in detail with further classification within kingdom Plantae, popularly known as plant kingdom. We must stress here that our understanding of plant kingdom has changed over time. Fungi and members of Monera and Protista having cell wall have now been excluded from Plantae, though earlier classifications placed them in the same kingdom. So the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter we will describe algae, bryophytes, pteridophytes, gymnosperms and angiosperms under Plantae. Let us also look at classification within angiosperms to understand some of the concerns that influence the classification systems. The earliest system of classification used only gross superficial morphological characters such as habitat, color, number and shape of leaves, etc. They were based mainly on vegetative characters or on the Andrewsian structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are most easily affected by environment. As against this, natural classification systems developed, which were based on natural affinities among the organisms and consider not only external fertilization but also internal fertilization like ultrastructure, anatomy, embryology and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification systems based on evolutionary relationships between various organisms are acceptable. This assumes that organisms belonging to same taxa have a common ancestor. We now use the information from many other sources to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all the observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome, number, structure, behavior and chemotaxonomy, that uses the chemical constituents of plants to resolve confusions, are also used by taxonomists these days. Coming into first type, that is algae. Algae are chlorophyll bearing simple thalloid autotrophic and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats, moist stone soils and wood. Some of them also occur in association with fungi called lichen and animals, example in sloth bear. The form and size of algae is highly variable, ranging from colonial forms like volvox and the filamentous forms like Elothrix and Spirogyra. A few of the marine forms such as kelps form massive plant bodies. The algae reproduce by vegetative, asexual and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being the zoospores. They are flagellated, motile and on germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. takes place through the fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix, or non-flagellated, non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous, and fusion of two gametes dissimilar in size as in species of Eudorina is termed as an isogamous. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed oogamous. Example, volvox and fucus. Here it is the picture of volvox. It is a picture of Eulothrix. It is the picture of brown algae, laminaria. This is the picture of fucus. This is the picture of dictyota. This is the picture of porphyra. This is a picture of polysiphonia. These two are types of green algae. These three are types of brown algae. These two are types of red algae. Algae are useful to man in variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy-rich compounds which form the basis of the food cycle of all aquatic animals. Many species of porphyra, laminaria and sorghum are among 70 species of marine algae used as food. Certain marine brown algae produce large amounts of hydrocolloids. Water holding substances. Example, algin, brown algae, and carrageen, red algae, which are used commercially. Agar, one of the commercial products obtained from Gelium and Gracilaria, are used to grow microbes and in preparations of ice creams and jellies. Chlorella, a unicellular alga-rich proteins, is used as food supplement even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae and Rhodophyceae. Firstly, we'll study about Chlorophyceae. The members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial or filamentous. They are usually grass green due to the dominance of pigments, chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate and cup-shaped, spiral or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation. Asexual reproduction is by flagellated zoospore produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous or oogamous. Some commonly found green algae are Chlamydomonas, volvox, Eulothrix, Spirogyra and Cara. Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous forms, ectocarpus, to profusely branched forms such as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A and C, carotenoids and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment, fucoxanthin present in them. Food is stored as complex carbohydrates, which may be in the form of laminarin or mannitol. The vegetative cells have cellulosic wall, usually covered on the outside by a gelatinous coating of algin. The protoplast contains, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to substratum by a holdfast and has a stalk. The stipe and leaf-like photosynthetic organ in the front. Vegetative reproduction takes place by fragmentation. Asexual reproduction is in most brown algae is by biflagellated zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the oogamous species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common forms are Ictocarpus, Dictyota, Laminaria, Sorghasum, and Fucus. The next type is Rhodophyceae. Members of Rhodophyceae are commonly called red algae because of the predominance of red pigment. Are physoerythrin in their body. Majority of the red algae are marine with greater concentrations found in warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depths in ocean where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in its structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization developments. The common members are Polysiphonia, Porphyra, Gracilaria, and Gelidium. Bryophytes. Bryophytes include the various mosses and liverworts that are found commonly in moist shaded areas in the hills. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession and on bare rocks or soil. The plant body
So here we will start from liverworts. The liverworts grow usually in moist, sandy habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of a liverwort is thalloid. For example, Mercantia. The thallus is dorsiventral and closely appressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of thalli or by formation of specialized structures called gemmi. Singular form gemma. Gemmi are green multicellular asexual buds which develop in small receptacles called gemmi cups located on the thalli. The gemmi become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into foot, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes, mosses. The predominant stage of the life cycle of a moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is creeping, green, branched, and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender, axis-bearing, spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding. In the secondary protonema, in sexual reproduction, the sex organs antheridia and archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte consisting of a fruit, seta, and capsule. The sporophyte in mosses is more elaborate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common example of mosses are Funaria, Polytrichum, and Sphagnum. Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissue, that are xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, sandy places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is a gametophytic plant body. However, in pteridophytes, the main plant body is sporophyte, which is differentiated into root, stem, and leaves. These organs possess well differentiated vascular tissue. The leaves in Pteridophyta are small microphyls as in Selaginella or large microphyls as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structures called strobili or cones. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous small but multicellular free living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, sandy places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophyte is limited and restricted to narrow geographical regions. The gametophyte bear male and female sex organs called antheridia and archegonia. Water is required for transfer of antherozoites to archegonia. The male gamete is released from antheridia to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular, well-differentiated sporophyte, which is the dominant phase of the pteridophyte. In majority of the pteridophytes, all the spores are of similar kinds. Such plants are called homosporous. But genera like Selaginella, Salvinia, which produce two kinds of spores, macro and microspores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophytes respectively. The female gametophytes in these plants are retained on the parent sporophytes for variable periods. The development of the zygote into young embryos take place within the female gametophytes. This event is precursor to the seed habit, considered an important step in evolution. The pteridophytes are further classified. The pteridophytes are also classified into four classes. These are Psilopsida, Lycopsida, Sphenopsida, and Pteropsida. Examples of Psilopsida are Psilotum, Lycopsida, Selaginella, Lycopodium, Sphenopsida, Equisetum, and Pteropsida, Diropteris and Pteris ediantum. Gymnosperms. The gymnosperms, gymnos, naked, sperma, seeds, are plants in which the ovules are not enclosed by any ovary wall and remain exposed. Both before and after fertilization, the seeds that develop post fertilization are not covered, and it means that they are naked. Gymnosperms include medium sized trees or tall trees and shrubs. One of the gymnosperm, the giant redwood tree sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza, like in Pinus, while in some others, like in Cycas, small specialized roots called coralloid roots are associated with nitrogen fixing cyanobacteria. The stems are unbranched, cycas or branched, in pinus or cedrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extremes of temperature, humidity, and wind. In coniferous, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata help to reduce water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form lax or compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspore develop into a male gametophytic generation which is highly reduced and is confined to only limited number of cells. This reduced gametophyte is called pollen grain. The development of pollen grain take place within the microsporangia. The cones bearing megasporophylls with ovules or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be born on the same tree, like in pinus. However, in cycas, male cones and megasporophylls are born on different trees. The megaspore mother cell is differentiated from one of the cells to the nucellus. The nucellus is protected by envelopes, and the composite structure is called an ovule. The ovules are borne on megasporophylls, which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form four megaspores. One of the megaspores enclosed within the megasporangium develops into a multicellular female gametophyte that bears two or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes and teratophytes, in gymnosperms, the male and the female gametophyte do not have an independent free living existence. They remain within the sporangia retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on megasporophylls. The pollen tube carrying the male gametes grows toward archegonia in the ovules and discharge their content near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and the ovules into seed. These seeds are not covered. Next topic is our angiosperms. Unlike the gymnosperm where the ovules are naked, in angiosperm or flowering plants, the pollen grains and ovules are developed in specialized structures called flowers. In angiosperm, the seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in wide range of habitats. They range in size from the smallest wolffia to tall trees of eucalyptus. They provide us with food, fodder, fuel, medicine, and several other commercially important products. They are divided into two classes, the dicotyledons and the monocotyledons. Our chapter is finished by angiosperm. So thanks.